Welcome to Straight Science, everyone. Straight Science is a science presentation series put on by UAF Northwest Campus and UAF Alaska Sea Grant, both here in Nome. And UAF Northwest Campus and myself, we serve the Bering Strait region, which is the homeland and waters of the Yupik, Inupiaq, and St. Lawrence Island Yupik peoples. Tonight we have part two. Part one was when, I think that was in March, when Michael Cameron, Josh, and Heather were part of a research cruise down up into the Bering Sea from Seattle with their Oscar Dyson, which we'll hear about. And they were off to do some research on ice-associated seals, and tonight we'll hear the results of that, plus some other interesting research ongoing. So tonight's speakers, there's three. They all hail from Seattle right now, so we appreciate you being on a different time zone and taking later in your evening to talk with us. And we have Michael Cameron, who leads the Polar Ecosystems Program at the NOAA Alaska Fisheries Science Center in Seattle, Josh London, and also Heather Zeal. And it looks like Michael Cameron's going to say a little bit about that. Do you want your questions as you go, or do you want to wait at the end since we have multiple speakers?
I think maybe waiting till the end would be best, if that's okay.
All right, we'll wait for questions at the end. And take it away, Michael Cameron. Thank you so much for coming.
All right, thank you, Gay. It's good to be back. As I mentioned, she mentioned, my name is Michael Cameron. I'm the leader of the Polar Ecosystems Program at NOAA's Marine Mammal Lab in Seattle. Some of you may know the name Peter Boving. Peter Boving used to be the leader of the Polar Ecosystems Program, but I've taken over. Peter's still in the program though, so spirit alive and well there. Josh London and Heather Zeal will also be presenting on some of their work a little later in the presentation. With all of us, we have many decades of experience working in Alaska with marine mammals and in other locations. The Polar Ecosystems Program monitors and studies bearded, ringed, spotted, and ribbon seals. Those are the ice-associated seals or ice seals. We also study harbor seals in Alaska. So all 5 phocids in Alaska, the true seals, are the ones that we support for the management and co-management. Seals in Alaska are monitored in 3 different ways. The first is the residents of coastal communities. Those folks are the eyes and the ears of those ecosystems, and often they're the first to detect changes in marine mammals and seabirds and other important resources. And especially hunters are frequently out observing the environment and the animals, and those observations are critical for understanding. The next way in which seals are monitored in Alaska is through the Alaska Department of Fish and Game. They work with hunters in order to collect samples that are harvested from Alaska Native hunters and to compile observations about the health and the status of those populations. The ADF&G analyzes things like reproductive success, growth rates, diet, disease, and contaminants. And they've also worked with various other communities, including the Ice Seal Committee, to show that the Alaska Native harvest of seals is at sustainable levels and currently not a threat to populations. The third way of monitoring is us. NOAA, the National Marine Fisheries Service, conducts broad area population surveys of seals in order to determine population abundance and trends. We also conduct a number of vessel-based tagging and village-based tagging programs within the breeding ranges. The work that we do at NOAA complements the observations of the hunters and also the work from the Alaska Department of Fish and Game because we tend to focus our investigations farther away from the coasts in areas that are less frequented by hunters. We also focus our work during the critical nursing, breeding, and molting periods. So those are the 3 main kinds of monitoring. They're all important because the seal populations are challenging to measure, and it's important to document them for detecting changes. Indications are, of course, that seals are responding to recent changes, especially in the Bering Sea, and so it's more important than ever to be monitoring them. There are a lot of important things that we want to monitor that can only really be measured in the spring, but that's the same time often that a lot of residents need to hunt marine mammals, but we believe that we can conduct these monitoring efforts, and we've shown that we can do them without impacting hunting success. One way we've been successful in that is because NOAA has a co-management agreement with the Alaska Native Ice Seal Committee. This is a group with representatives from the 5 boroughs that are at harvest ice-associated seals in Alaska. The Ice Seal Committee has a working group that also, in addition to NOAA, also includes the Alaska Department of Fish and Game and educational and other institutions. And together that group develops a Alaska ice seal research plan every year or every other year. In that plan are identified all of the projects that folks either have recently conducted or would like to still be performed in Alaska as a way of sort of focusing the research that can help inform co-management. And all of the projects we'll be talking about today were either have been or are currently still included in that ice seal research plan. The first project, I'll just give a quick background, is our large area aerial surveys for ice-associated seals. Our goals are to determine the abundance and distribution of seals and polar bears throughout Alaska. Polar bears is a cooperative work that we're doing with the U.S. Fish and Wildlife Service. It's an international effort in order to survey the sea ice habitat of the Bering, Chukchi, and Beaufort Seas roughly every decade or dozen years. We completed our first survey of the Bering Sea in 2012 and 13, and we hope to be back again in 2024. I'll mention that the international part is that whenever we conduct work in the Bering or the Chukchi Sea, we try to coordinate our efforts with our Russian colleagues who are also conducting surveys at the same time using the same methods. Similarly, when we do work in the Beaufort Sea, we try and have some coordination with our Canadian counterparts so that we can have better information for the total populations. Those surveys are conducted using a manned aircraft, a NOAA Twin Otter. We fly high, which allows us to reduce disturbance to animals on the ground. And we fly fast so that we can cover large areas in a relatively short amount of time. We employ an instrument-based approach rather than folks looking out the windows and counting or taking photos. We have a camera suite of combinations of thermal, color, and ultraviolet cameras or sensors that allow us to locate seals, thermal signature, for example, in the image below, and then identify them to species with the color images. Our discussions, again, with the Alaska Native Ice Seal Committee, coastal communities, whaling captains, and et cetera, have helped us to avoid impacting hunting activities by being able to help direct timings of our surveys and locations. I won't be going too much into the abundance and distribution work for this talk. I just wanted to give a brief background. These are then the current abundance estimates for the 4 species of ice-associated seals in the eastern Bering Sea. This is just for the US side, and for the Chukchi Sea. We only just completed our surveys for ice-associated seals in the Beaufort Sea, so we don't have estimates for those yet. But the main thing I wanted to talk today to you about is our sample and tagging work at the sea ice in the Bering Spring in the spring, excuse me. For this work, we want to understand how many seals there are in the populations, their seasonal movements, how healthy they are, and how they're responding to habitat changes. In order to do that, we have to capture the animals. In order to do that, we have to get close to the animals, which requires us to move to the southern edge of the Bering Sea pack ice in spring. Since 2005, from about 2005 to 2010, we've been conducting this work in May or June. Starting in 2014 and for every other year after that, we've been focusing it more in April. different type of research focusing more on moms and their newborn pups in April. Unfortunately, in 2020, we were unable to conduct the work because of COVID. But in 2022, just last month, we were able to complete some work, and that's what we'll be talking about today. We use the Oscar Dyson to get access to the marginal ice zone of the Bering Sea. We use it, we have a number of small inflatable boats on board that will launch and then move into the pack ice, quietly moving through the pack ice, communicating with one another, looking for ice-associated seals to capture. Once we do that, we coordinate, slowly moving towards the animals until we can jump out onto the ice floe where the seals are hauled out and capture them using handheld nets in the way that you see here. At this time of year and in this location, we tend to mostly capture ribbon and spotted seals, although we have captured the occasional bearded and ring seal for our work. Once an animal is captured is when the work of sampling and tagging can happen. We instrument animals with satellite linked tags, and that's for information on movements, foraging behavior, and hauling out. Josh London will be talking about that in just a minute. We also collect information, measurements, tissue samples, and conduct estimates of the seal's health. Heather will be referring to that after Josh. And then finally, as a pilot project in 2022, we were starting to use unmanned or uncrewed aerial systems to help us investigate body condition in a different way. As I mentioned, we don't want to be impacting the hunt or hunters. We don't conduct any of our work within 12 nautical miles of the coast or within 30 nautical miles of any whaling or sealing or hunting communities. We also maintain daily communications while we're out there by sending emails of our location and our activities for that day, our plans of the day. In 2022, I sent out 200 emails a day, or I sent emails to 200 people with these sort of daily updates. If you're interested in being put on that mailing list, just let me know after this, or Gay, and we'll make sure that you're on the mailing list for this sort of work going forward. We always work to avoid whales, walrus, and hunters whenever we see them, if that's ever something we'll see. It's important to remember also that, like hunters, we don't want to disturb the animals that we're studying. Capturing requires stealth and quiet, and disturbance will be impacting our monitoring. And also, the Oscar Dyson, though it is a large ship, it is designed to be acoustically quiet. Specifically underwater noise, in order to avoid disturbing fish for fisheries research, which is what the Oscar Dyson is mostly conducting. So in 2022, our expedition aboard the Oscar Dyson was shorter than most of our other expeditions, just 18 days versus our usual 28 days. And from that, we only had 11 days at the ice zone, which is about half of what we normally get. During those 11 days, we had rather unusual weather and sea ice conditions. Heavy fog, snow, high winds, and swell made it difficult for us to be able to see seals from the bridge of the ship to capture them or to find them. Perhaps also more importantly, there was a cold snap in February in the area, which formed a lot of new ice south of the thicker ice that tends to be preferred by ice-associated seals. What this meant was that it was difficult for us to get access to some of the habitat that we expect a lot of these ice-associated seals to be preferring. As such, we didn't see very many seals during this cruise, less than 30 for the entire expedition, despite being in areas near the shelf break or in shallower waters, in areas that in the past we have been seeing a lot more animals. One difference, though, from previous cruises, we used sonobuoys aboard this cruise, which are essentially underwater microphones that allowed us to listen for seals and other marine mammals. And we did hear quite a lot of activity under the water, no matter when we put the sonobuoy in the water. So it was interesting that even though looking out the window, we weren't seeing much, we were still hearing quite a lot. This is the general area just west of St. Matthew Island, where we tend to conduct most of our work and where we were working in 2022. These red lines to the left just indicate the bathymetric isobath. So this just determines water depth at those different locations. This is an ice image, a radar sat image that can look through clouds from April 10, about the time that we started our work. This is just my tracing, so it's not the best job, but it sort of shows, you can tell from the image which ice is new or young and thin, of the type that doesn't tend to be preferred by ice-associated seals at this time, who like to have thicker, older ice in order to give birth to their young and have a stable platform for nursing. About a week later, there was a rather large storm that lasted for a few days and tended to blow a lot of that ice, the younger, thinner ice away, which gave us access to some of the thicker ice that we were hoping for. It was around this time we managed to capture our first seals. And this is what the sea ice looked like around the time that we finished. I apologize, I think now you might not be able to see the slight green line. That shows our actual track line of where the ship went for the whole time we were up there. So at this time, I'm going to transfer the presentation over to Josh London, who's going to be talking a little bit more about our satellite tagging operations. Thanks.
Thanks, Mike. I'm really happy to be here tonight and to share with you guys some of the, not only some of the information about the work that we just did, but give you maybe a bit of a deeper dive into some of the data we've gotten from the biologgers, or the satellite tags that Mike referenced over the more than a decade now that we've been doing some of this work. And so I thought we'd just take an opportunity to share some of that information and then tie that into some of the work that we just did. The one thing I want to do here quickly is for those of you who are interested, this presentation is available on the website as well. So if you want to check it out later on, I just pasted that into the chat. So I also realized this is probably one of the first times I've ever given a talk to folks in Nome or the Northern Bering area. And so some of you may not know me. Just want to take a quick moment and introduce myself. As Mike said, I have been with the Marine Mammal Lab in Seattle for almost 15 years now. But originally I'm from Tulsa, Oklahoma. That's where I grew up. And I moved to Seattle in 1993, went to the University of Washington and stayed there and did my PhD, my graduate work, studying seals, salmon, and killer whales here in the Seattle area. That same time I met my wife, Shanna. She's actually originally from Mexico City and moved to Seattle about the same time. And we've happily lived here in Seattle for many years. Of course, I thoroughly enjoy all the opportunities I get to come up to Alaska and to meet folks in the communities there. And then I'll also point out that we have our dog Barley, who is originally from Northern California, but she's been with us here in Seattle since 2019. So just want to take a moment and introduce myself. I'm always excited to talk about seals in Alaska. Feel free to reach out via email anytime if you have questions. And I hope this is the first of many opportunities to connect with many of you. So let's talk about what are biologgers. You know, Mike mentioned their satellite tags. You'll hear different kind of names for them, but generally you can think of them as kind of smartphones or smartwatches for seals. Just like your phone, you know, the biologgers know where they are, they know location, but they also track information. So, you know, maybe some of you are tracking how many steps you get every day or how many miles you run, how fast you run, what your pace is, what your heart rate is, all that kind of stuff. Similarly, we can attach these types of devices to seals and then we get some information back about where the seals go and what their behavior is. And that's essentially what these devices are. But unlike your phone or your smartwatch, we're kind of asking a bit more of these devices because we need them to operate in some pretty harsh conditions. You know, very cold temperatures in the Arctic when the seals are out of the water and on the ice. In the water, they're diving deep to 600, 700, 800 meters. And that device has to be able to withstand those pressures over and over and over again. Battery life is limited. If you think about it, maybe you'll get 1 or 2 days of charge from your phone, but you have the option to, you know, find an outlet and plug in and recharge overnight, plug into your car when you're driving, things like that. We don't really have that option. The seals don't have a little outlet to plug in when they haul out on the ice floe. And similarly, they don't have Wi-Fi either to kind of share that information with us. So we have to rely on satellite transmissions in order to get the information back. So these are actually pretty amazing engineered devices that we're able to learn so much about seals from devices that are so robust and can handle these really harsh conditions and do that in a pretty short, with a short battery life. That's pretty amazing. On the right side, we have 2 kinds of tags that we tend to deploy in our seals. One in the upper right are tags that are actually attached to the interdigital webbing of the rear flipper. Those are great because they stay on for a longer time. And then the tag in the lower right, that tag is glued to the seal's hair. So here again on the right, you can see the tag that's glued to the hair. And on the lower right is the tag attached to the interdigital webbing of the flipper. The tag that's attached to the hair, every spring the seal molts, and so that hair falls out and the tag falls off with it. So Mike talked some about the timing of our cruise. When we're doing this cruise now in April, we get fairly short duration of our tags because the seals are about to enter their molt. Their hair is, you know, 10, 11 months old. So maybe not as strong as it was when it was first grown. But when we did some of the work early on, and we were deploying tags in May and June, the deployments would last 9, 10 months. And so later on, I'll show you some data that where we have information from the entire year to give us some insight into what seals are doing. The rear flippers are great because they don't fall off. They can stay on for a couple of years. We've had some bearded seals with tags on for 3 to 4 years. It's a similar approach. ADF&G and North Slope Burrow also use these tags. And we've really gotten some great insight into seal behavior long-term, year after year, that we wouldn't have gotten with any of the other types of tags. I also wanted to take a moment and just kind of talk a little bit about the ethics and the humaneness of attaching biologgers and devices to seals. And actually this goes for everything that we do in our research program. We've kind of 2 laws that kind of guide the work that we do. The Marine Mammal Protection Act, we have to get a research permit. And actually it may seem kind of odd, but the National Marine Fisheries Service actually issues a research permit to us who work for the National Marine Fisheries Service to do our research. And that's part of the law, and it's part of our code of research where we are, you know, describe what we do, and then that permit and the proposal of the various work is reviewed by experts and also out for public comment. And then in addition to the research permit, we also have to work under what the U.S. Animal Welfare Act, and there's an institutional or an IACUC, you may have heard of, an animal care committee that reviews our work. And they really, it's made up of vets and other experts who really look at our proposal and look at the work we do, look at our history, and make sure that the work we do is humane. And the key principle that all of this is that we're trying to maximize the information that we gain with the least amount of impact to the seal. So Some of what I'll talk about is all the information we get back from these devices, and that's what's really great is we can fit into a relatively small package a lot of sensors that give us a lot of information so that we maximize what we get back for the least amount of impact to the seal. So the first kind of area that I'll talk about that we get from these devices is movement and space use. Where do seals go? The locations we get from the satellite transmissions and GPS data that's stored on board. Sometimes the locations aren't great. They can be within a quarter mile or more. But we get about 8 to 10 locations a day. And for seals in an area like the Northern Bering Sea, it's a really great information on understanding where seals go. We're asking questions of how far do seals range in the North Pacific? Where might they forage? Are there favorite oceanographic features such as temperature or bathymetry that the seals like? There's also interest in understanding how where seals go, where they feed, how that might overlap with commercial fisheries or shipping traffic, exploration activities. And then as we're all thinking about just how much the environment's changing, especially the springtime and the Bering Sea, we want to understand kind of how these seals might respond over time to these changing conditions. And this is just one example of the kind of information we can get. This is some of the early work that we did where I mentioned that we get deployments of seals, deployments of tags on seals throughout the year. And what that allows us to do is to see how the ribboned and spotted seals change their distribution throughout the year. So what I'm showing you here is the kind of November to February, that winter period. You can see on the left side are the ribbon seals and on the right side are spotted seals. And you see how ribbon seals are a little bit more spread out through the Aleutians over towards Kodiak, over on the Russian coast into the deeper parts of the Bering Sea, whereas the spotted seals tend to stay on the shelf and shallower waters. But then as we move into the spring, both ribbon and spotted seals really concentrate their movements in that area where we were trying to catch those seals. That kind of shelf break and the southern edge of the Bering Sea ice zone. And then there's an even greater expansion when we get into the open water summer period. And you can see a lot of ribbon seal movement on the left side, over into the Gulf of Alaska, up into the north of the Chukchi into the Arctic. Whereas there's a bit more movement of spotted seals moving a bit more coastal, but staying kind of on that shelf break. And so this is the kind of information we can get when we kind of pool all this information together, all the data from the tags of where seals are, and we start to understand differences between ribbon seals and spotted seals in this really big kind of spatial scale. Dive behavior is another aspect of these tags that I think is really fascinating. Each tag has a pressure sensor on it that records depth every second during the deployment. So it's just recording depth all the time and logging that to the tag. The tag then will do some math and calculations on board to summarize that behavior and to time at depth over a time period. So how much time is the seal spending at different depths within, say, a 6-hour period? And then when the tag comes to the surface, it bundles that up into a little message and sends, imagine like a quick email telling us for that period what the seal was doing underwater. And what that allows us to look at is, how often do seals dive? How deep do they dive? Are there important depths for foraging? Do seals go to the bottom or do the seals forage at the midwater? How might their dive behavior change across seasons? How do young seals learn to dive? You can imagine, seals when they're born don't really know how to dive, and it takes a while for them to kind of figure it out. And then the physiological challenges. As you'll see, some of these seals are diving extremely deep and starting to think about and understand how they overcome those challenges is quite interesting. So this is a kind of a big pool. I just put all the data that we have for ribbon seals from 2005 to 2016. It's tens and tens of different deployments. And for every day of the year, just kind of figured out what was the median percent time at these different depths that the seals we had biologgers on were diving to. So you can see there's some cycle. There are periods of time when ribbon seals are relatively shallow only, 150 meters or so. But there are other times throughout the year when ribbon seals are diving down to 600 meters or more. And that's a long ways for a small seal to go down and back. But they managed to do that in about 8-10 minutes, sometimes 12 minutes for a longer dive. And so we get some information about how these seals use their environment. And like Mike was saying, we were out looking out above water on the ice and not seeing many seals, but the sonar buoys were telling us that there were seals underwater. This is telling us that seals are really active underwater. There's lots of stuff that they're doing. They're diving into different depths, and that changes over time as they move throughout that environment. The interesting thing, just like before, we showed that ribbon seals kind of had a broader range in their space use compared to spotted seals. The same is true for dive behavior. There was a few times when in the spring, when spotted seals are closer to the shelf break, when they have access to deeper water, they definitely seem to take advantage of it. But a lot of the time, spotted seals are spending on the shelf break where it's 50 meters, 80 meters, something like that, and spending a lot of time on those shallower waters. And so we don't see the deep dives from spotted seals that we do on a regular basis from ribbon seals. This is some data just fresh from this particular cruise. We had the opportunity to look at a finer scale information about a ribbon seal mother with a pup that we deployed a tag on. And the interesting thing here is that when we released the seal, she had her pup with her and she was nursing. And so what's interesting is that for these 5 days that I'm showing here, you see these periods where the gray bars indicate when the seal was out of the water resting on ice. So she was probably nursing her pup, but then would do a little bit of foraging and then come back, a little bit more foraging and come back, and a little bit more foraging and then back to nurse. So we think that mom is nursing her pup, but then going off and getting a little food to sustain herself, coming back, doing some more nursing, maybe moving the pup from one flow to another. The ice is quite dynamic, and it's not surprising that the mom and the pup would need to move to a couple of different ice flows over time. But it's really interesting to think about, a ribbon seal mom, and that she's got to nurse her pup. She's going to leave that pup on the flow, go dive, get some food for herself, and then come back and continue nursing. It's a pretty amazing feat. And we're just starting to get some information about this from some of these recent tags that we've deployed. Haul out behavior is another thing that's really important to us. There's a saltwater sensor on the tags that tells us when the tag's dry versus when it's wet. And so we get the percentage of each hour that the tag is dry. It's a key product that we get from the flipper tags. The graph on the right, you can see the orange areas are times when the tag was dry. The blue areas are times when the tag was wet. And this is one of those flipper tags. So we actually got 2 years from this ribbon seal in 2016 and 2017. And you can see it's in that April-May period when ribbon seals are spending more time hauled out, whereas early in the spring, they're spending less time hauled out. And that kind of data is important for us because we want to know how important is sea ice for seals to rest when they're out of the water. For the aerial surveys that Mike talked about, we want to know what portion of seals are hauled out versus what portion are in the water, so that we can get a better estimate of population size. Maybe there are differences between age and sex classes. And then at other times of the year, maybe we can get some indication if seals are choosing to haul out on land versus sea ice. And when we put all that information that we have together, similar with the dive data, I can bring it together and build a model of haul out behavior for ribbon seals and spotted seals. And the yellowish orange areas tell us times of year and times of day when seals are more likely to be hauled out. And so in general, you can see that ribbon seals and spotted seals in the springtime really like to be, are more likely to be hauled out around the middle of the day, local solar noon, and less likely, and maybe in the evenings, You also see that there's early in the spring, they're less hauled out. And then there's this period when they're really focused on nursing, on breeding, and then on molting when they spend more time out of the water. The last thing I'm going to talk about is some new tags that we put out this year that allowed us to start to look at oceanographic data. So in this case, the seals are actually doing us a favor. They're collecting information on water temperature at different depths and then transmitting that back. And that's information that maybe oceanographers couldn't get because the seals are going places they don't, like say into deeper ice or things like that. And this is something we hope to expand more so that as more seals are out there collecting this information, we can provide more information to the greater community about the Bering Sea and how it's changing with respect to oceanography. And so just as one example, these are a couple of the different temperature profiles that were provided by our two seals that I just had a chance to take a look at. And the interesting thing here is you can see that both seals, when they're in shallow depths, the water's colder. So that's kind of typical. There's sea ice. And so you've got cold water at the surface, but then as the seal dives deeper, the water temperature actually warms up. And this thermocline is an important oceanographic feature that moves up and down and changes with the season. And so we hope with more of these tags put out, our seals can help oceanographers understand better what's going on with the ocean and how it changes. And we're really looking forward to that opportunity in the future. So that's all the information I have about biologgers, but now I believe it's time to turn it over to Heather and let her talk to you about some of the tissue samples and stuff that we collect.
All right, take it away, Heather. And if everybody, I guess we're holding our questions to the end. I know I've got a couple and we'll wait till the end. All right, we can see you, Heather. Fire it off. Thank you.
Okay, great. Thanks, Josh. All of that is so interesting and such new stuff too, just from these tags this year. Really cool. So hi, everybody. Thanks for coming and listening to our talk. Really glad to be here. So my name is Heather Zeal, and I've worked for the Polar Ecosystems Program since 2007, working with seals in Alaska on various studies. But before starting work with marine mammals, I worked in Maine studying genetics of an upland game bird, and I did some work on the Columbia River in eastern Washington with fish and seabirds, doing an avian predation project. So Alaska was new when I started here, but it's been wonderful. So Mike gave an overview of all of the projects and research that Polar has been doing, and then we just heard about all of the telemetry from Josh. So now I'm going to give a little bit more detail about the sampling that we do while we're in the field and some of the projects that we have going on using these samples to monitor and look at the health and condition of the seals. So after we've caught the seals, which Mike explained, we take a bunch of samples and we start generally with morphometrics. So we take mass, length, and girth measurements. And the picture on the left shows how we get weights from seals when we're out on the ice floes. So we take this bipod out, and luckily it breaks down into smaller pieces so we can fit it into the small boats that we use to go capture the seals. And then we've got a hanging scale and a sling with these straps. So we get the seal into the sling, attach the straps to the scale, and then use the bipod to lift it up. So it works really well, and we can get it all out onto the ice. And we also take lengths. We take standard and curvilinear length, all while the seal is in a prone position, as shown in this picture on the right. And we also take axillary girth, which is measured just behind the front flippers, and maximum girth and hip girth. We also collect blood from all the animals. We use several types of tubes to collect blood, and then we use the different parts of the blood in several studies. We also collect tissue samples from every animal. We collect 2 samples from each animal. When we attach a satellite flipper tag, like Josh was describing, we collect the biopsy punches that are taken from the flippers before the satellite tag is attached. If we don't attach a flipper satellite tag for some reason, if it's a pup or another reason, we just take little snips from the flipper webbing for genetics. And we've done this for every animal that we've captured, so we have a good archive of tissue samples for genetics projects. We also take nasal and rectal swabs, and these are generally used for a variety of disease analyses and screening. And the number of swabs we take depends on studies that need them or are asking for them. So for example, we always take 2 nasal and 2 rectal swabs that are used for sort of general disease screening. But this year when we were out, we also took swabs that were specifically assigned to, or they're earmarked for analyses for the UME and for COVID analysis. We also take a whisker. We collect a whisker from every animal, and we use these needle nose type pliers to pull a whisker. We try to get the longest whisker on the left side and get the entire thing, including the root. And the whiskers are being used for stable isotope analysis. We also collect hair. The top picture shows how we usually collect hair. We use an electric razor and shave a little patch to take all of that hair. But the bottom picture shows an animal that's molting. So if we are lucky enough to catch a molting animal, we'll collect both the old molting hair and then shave a patch of the new hair. And so that gives us some good information about different timelines from when the hair has been growing. So both the old molting and the new. We collect scat, any scats that we get. And the scats are primarily used for information about food habits. We take the scats back to the lab and clean them and then collect the hard parts, so the bones and the otoliths. And then we can identify prey that they've been eating from those hard parts. And we've also used scats, and we'll use them for some additional projects as well. We have an ultrasound, and we use the ultrasound to measure blubber thickness of the animals, which can be used as a measure of body condition. We take measurements of blubber depth at 4 locations on the seals. So we measure on the dorsal side at the axillary line, so behind the front flippers, and at the hips. And then we measure on their right side, on the right lateral side, also at the axillary and the hips. So that was a quick overview of samples we collect and how we collect them. And this is a list of some of the research projects that those samples that we collect have been used in. And I will go over some of these projects also. So we screen for a wide range of pathogens as part of our monitoring program. We analyze the serum from the blood and nasal swabs to look for pathogen exposure and possible infection. And so the purpose of this screening is mainly to establish baseline data about pathogen and disease in the seals. So we want to determine the current numbers so we can monitor changes over time. And this is particularly important now at this time with these rapid changes in the environment, as changes, these rapid changes could affect both survival and distribution of both the pathogens and the seals. So getting a picture of what's going on right now with disease in seals is really important. And we found in the samples that we've analyzed that the seals have been positive for exposure to a few of the pathogens we've tested for. And this just means that the seals that we tested were positive for antibodies to that pathogen. And I know everybody is familiar with antibodies now with COVID. So it really just means that the seals were exposed to that pathogen and they've survived. We also have used blood to collect hematology and serum chemistry data. We got a vet scan in a few years ago, so we were able to get hematology data for all of the seals that we capture. We don't have a way to get serum chemistry data yet, but in 2016, we worked with Colleen Reichmuth at UCSC, and we were able to get samples from spotted, ribbon, and bearded seals analyzed for serum chemistry. So we have results from those animals. And then in 2019, this paper was published with Colleen and Carrie Gertz from the Alaska Sea Life Center. And in the paper, they compared values of several health parameters, including hematology and serum chemistry. They compared those among captive seals, seals that were in a short-term rehab situation, and then wild-caught seals. So the results and the ranges that were published in this paper from the wild-caught seals were from samples that we were able to collect in the Bering and out of Kotzebue for the bearded seals. And other samples that we're using, so we have skin snips, like I said, for every animal. We haven't actually started a big long-term genetic study for ice seals, but they're all archived, so we'll be able to use them for studies on population structure, gene flow, genetic diversity in these ice seal populations. We're analyzing hair samples, or we're working with Lori Rhea, and her lab is analyzing hair samples for mercury content at University of Alaska Fairbanks. So we'll be able to get an idea of contaminant exposure in these samples collected from the middle of the Bering Sea. And Lori's lab is also analyzing whisker and hair samples and doing stable isotope analysis for those samples. And the stable isotope data will give us information about feeding ecology of the seals. So we can get some information about
Looks like Heather, I don't know if you can hear us, but you've certainly frozen. Is
I
that
can
the
hear
internet?
you. I think she's frozen. I'll see if I can get her really quick. Otherwise,
Am I
might
back?
have some of her. Oh,
You're
there you're
back,
back again.
Heather.
Am I back? Okay.
You're
Yeah.
back.
I think that was a VPN problem.
Okay.
I think VPN just cut me off. Can you still see my screen?
No,
We're seeing you.
Okay.
which is good. I mean, you
Okay.
crawl back on with your screen, but tough having those
There you
that
go.
internet in Seattle.
Am I back?
You are.
You're good.
Sorry about
Continue
that.
on.
VPN. Okay, so I think I was talking about stable isotope analysis and how that gives us information about about feeding ecology. So we also, I didn't mention this since I didn't have a picture, but we provided urine samples from ribbon and spotted seals that we collected on our cruises. And we provided them for this study that Kathy LaFay published in 2016 on algal toxins in Alaska. And they were looking for domoic acid and saxitoxin in these 13 species of marine mammals. So I don't know if you can see my arrow, but these are the spotted and ribbon seal samples that we were able to provide. And those were positive for domoic acid. And on this recent trip, we were collecting ice. She wanted us to collect ice algae. So we've got ice algae samples for another study that she's working on. So now I'm going to talk about some of the body condition work that we've done using the samples and data that we've collected in the field. So this slide shows sea ice in the Bering Sea in April in 2012 and 2018. And so in April, these ice-covered areas of the Bering Sea are crucial reproductive areas for populations of ice seals. So any reduction in these ice-covered areas then likely has implications for all the populations of ice seals in the Bering. You can see that in 2018, it was an extremely low ice year. And we actually had a capture cruise that year, which Mike mentioned. And you can see, so this orange circle indicates seals that were captured in 2007 through 2016. And the yellow circles show the locations of seals that were captured in 2018. And you also saw that we used the Oscar Dyson to get up there, and the Dyson can only skirt the very edge of the pack ice. So you can see how much farther up that marginal edge of the pack ice was in 2018. So also in 2018, an unusual mortality event was declared for bearded, ringed, and spotted seals. This was declared because starting in June of 2018, bearded, ringed, spotted seals started showing up in really large, much larger numbers than typical, and that's shown in these with this yellow and orange lines in this figure, and they were showing up all in the Bering and Chukchi Seas. You can see in this map, and these numbers were 5 to 7 times the yearly average from 2000 to 2017. So NOAA declared a UME. In 2020 though, the numbers of the dead seals returned to near average, and sea ice was partially rebounded. There was definitely more ice in 2020 compared to 2019. So the other potential effects due to loss of sea ice for ice seals could be poor pup production due to the lack of ice platform, poor pup survival due to less area for resting and nursing, and poor nutrition and body condition due to ecosystem effects on their prey. And we focused on this last point and asked this question: are there trends in body condition of seals in Alaska that might reflect ecosystem changes, like impacts on seals' prey during recent rapid environmental change? So we used data and measurements that we've collected in the field to come up with 2 measures and indexes of body condition. The first one was using morphometric data. So we used mass over standard length as an index for body condition. And the second was using blubber thickness measurements that were taken from the ultrasounds as an index of body condition. We ended up deciding to use the morphometrics of mass over length as an index instead of blubber thickness, mainly because we only started collecting ultrasound data in 2014, but we've collected morphometrics since 2007. So we had a much larger sample size. And for these analyses of body condition, we also included harbor seals that were captured in the Aleutian Islands. So this is a map here. This shows the map of locations where we captured ribbon and spotted seals that were measured for this study. You can see again, these are the locations of seals captured in 2007 through 2016. And then these are the locations of seals from 2018. And the triangles are the locations of the harbor seals captured in the Aleutian Islands. So using the morphometrics of mass over length as the index, we were able to have, we got a sample size of 94 spotted seals and 98 ribbon seals. We also included 80 harbor seals in that analysis. So using these data, we published this paper in 2020 with the analyses that use the index of body condition, this index of body condition of mass over length, to look at the question of whether and how trends in seal body condition are related to recent the rapid changes in the environment. And so here's some of the results from the analyses in the paper. We found that body condition of ribbon seals of all ages and both sexes and body condition of spotted seal pups declined from 2007 to 2018. So which that means the body condition of pups of both ice seal species declined, suggesting that maybe poor nutrition for mothers during gestation or nursing or both affected body condition. There's one possible explanation. We also found that body condition of subadults and adults declined in ribbon seals, but not in spotted seals. And so when the April ice edge is so far from the shelf break, it's like it could be that ribbon seals might not be able to reach their preferred deep water foraging habitat off of the shelf. So that's another potential explanation of why we saw the body condition declined in ribbon seals, but not spotted seals. So using these indexes, using this index of body condition, and we answered basically that yes, we found that trends in body condition did appear to reflect changes of this most recent rapid changes in the environment. But we still have the question of why, and I'm going to give it back to Mike to talk more about some of the projects that we're still working on to try to answer some of these questions on body condition. Thanks.
Thanks, Heather. Let's see if I can do this. Didn't work. Are you seeing my screen?
Not yet.
No,
We were
no.
seeing ourselves. Here we go. This is looks
There
like,
we go.
Nope.
Let's do this. Nope.
There.
Okay.
You got it.
All right. So scientists are greedy. We need more data. So capturing seals in order to weigh and measure them clearly, we've shown that to be useful. It's also work that the Alaska Department of Fish and Game has done using harvest collected samples. But it's difficult to collect a lot of samples from animals, live animals far from the coast. And so in order to increase that sample size, in order to be able to make better inferences, we need to find other ways of being able to collect information like the body condition statistic that we developed. We recognize that other researchers had used uncrewed aerial systems, or UAS, with cameras in order to measure animals and estimate mass. And so we decided to try our hand with that. So again, the goal then is to estimate the seal's mass and the seal's length. Those are the two components of our body conditions index. From a seal that's 100 foot away from where we are, we can't touch it, and we can only use it from a photograph. Turns out that length is a fairly easy thing to measure if you know the characteristics of the camera and lens you're using, and you know the distance to the target. You can identify the dimension of each pixel in an image at ground level. So as long as you know your altitude and your camera, you can just count the number of pixels and get a length from an image. Mass is a little bit more complicated. Mass we tend to infer as a function of volume. That is, what is the volume of the seal? So in order to estimate volume, we need to be able to also use those measurements. Now, the relationship between those measurements and volume and those measurements and mass require additional data. And the best way to get those data is to have animals that will do what you want them to do when you want them to do it. And to do that, we cooperated with researchers at the Long Marine Lab, UCSC Santa Cruz, and the Alaska Sea Life Center. This is Colleen Reichmuth here. At these labs, they have captive bearded, ringed, and spotted seals that are trained to hold positions and that are frequently measured and weighed. Using these animals at different times of year, we were able to and will continue to conduct some sample flights with the unmanned aircraft over these animals to get measurements from the air, and then be able to compare those to the actual ground truth estimates of seals that we can weigh with a scale and weigh with a tape measure, or excuse me, measure with a tape measure, in order to improve our statistical models to combine, to be able to use measurements to estimate volume, to estimate mass. We do these samples at 2 different times of year. Once in the summer when the seals are at their leanest, and once in the late fall when the seals are at their fattest. We do this such that we hope to be able to, ultimately, of course, we hope to be able to detect changes in population and body condition. So to be able to actually look at changes in body condition, seasonal changes for the same individuals will give us a good idea of how well our statistical models and our methods are working. This is just an example of the type of image that we were taking from those captive care facilities. This one happens to be at the Long Marine Lab. Taking an image of a known altitude with other scale bars for reference, we can be able to get good estimates from this animal. So we will be continuing to collect those data at the Alaska Sea Life Center again here in June and again in September. But as I mentioned, we wanted to do some tests to see how well we'd be able to take images of seals off of the, actually in the wild. These are animals that are hauled out on sea ice. So we did some practice flights using an unmanned aircraft from our work off of the Oscar Dyson. As I mentioned, however, we saw very few seals during this cruise, and so we really had very few opportunities to test the system. We did have some issues being able to launch the ship when we were standing on ice floes, or the ship itself when we were in heavy swell, but eventually we learned how to overcome those issues. One problem we thought we might have would be with the weather, temperatures of the wind, perhaps icing. Those didn't seem to be an issue. So from a functional standpoint, once we were able to work out the bugs, that worked pretty well. These unmanned aircraft also have a thermal imager that allows us to locate seals, which we expect will also be helpful when we kind of expand this project to a more larger sample. Being able to locate seals on the ice is helpful. This is an example of a thermal image of a mom and her seal pup. You'll notice the seal pup I think you can see my cursor here. I should hide this. The seal pup here, you can barely see, but that's because that seal pup just recently came out of the water and is still cold, unlike the mom, who is warm. At the same time, this is the color image that was taken of that same mother pup pair. And as we get closer, we can zoom in. And again, we know our altitude. We can then count pixels and get a good estimate. We can also count the area, that is the footprint, the silhouette of the seal, and perhaps use that as an index. And then, of course, use those measurements to be able to estimate volume, relate that to mass using the data that we're collecting from the Alaska Sea Life Center and the Long Marine Lab. As I mentioned, biologists are greedy by nature. We always need more data. We did not get a lot of seal data on this cruise because, for the reasons that I mentioned. So we needed to find other things to measure on the ice. In this case, we used our statistician. This is Paul Kahn, whom we've also decided now to get his body condition statistic. Anything we can do to increase our sample size, I suppose he appreciates, although he probably wasn't expecting this. That's the end of the presentation of the work that we do and the Polar Ecosystems Program, and more specifically the sort of preliminary, early, fresh, hot off the press results from our 2020 Dyson cruise. I wanted to bring this to your attention, however. Every year, the NOAA Fisheries, Alaska Fisheries Science Center, our group, publishes this map or this flyer of all of the field work that's expected to happen around Alaska on marine mammals. You can locate it at this URL that's up top. For each one of the projects that's listed along this side, there's also a more detailed research brief. This happens to be the research brief for our project. I'm just bringing this up because this is all really useful information if you'd like to know all the work that's happening in your backyard and find good contacts for people to talk to about if you would like to know more about their research. So I really appreciate the opportunity to today to be able to talk to you about this. We find our work very exciting and we love sharing it with people who we hope also find it exciting. So thank you very much. And if there's nothing else, I think we can have some questions.
All right. Well, thank you very much, Michael, Josh, and Heather. Holy cow, that was a lot of information. And this is the time we will do questions now. And this is the time for the street science crowd to give Michael, Josh, and Heather a little love in the chat box. Not easy to be a street science speaker, and they certainly gave us plenty to think about tonight. So, and we really appreciate it. Your slides were beautiful to look at as well. Can't go wrong with a ribbon seal picture, my goodness. But all good. So while people are writing in their questions, are there any burning questions, right? Are writing in there, giving some love. Are there any burning questions? You can either type them in the chat or you can, if you're like a Zoom aficionado, you can raise your hand, but or you can just blurt it out. So while people are thinking about that and writing in the chat, my question is, you know, there's a lot of interest this year. When you guys were out there, it was stormy, but up here, our ice was disintegrating. And I know you guys had a delay getting out, and then you got hit with that weather and whatnot. But my curiosity is, what would be the most unexpected thing you saw during that time? Because we are seeing that at that time, the ice was really unexpectedly, for a lot of people, it was unexpected to see it disintegrate so quickly. Did you guys see anything really unexpected on your travels out there? You were really down off west of St. Matthew. That's a pretty interesting place to be at that time of year for lots of reasons beyond seals.
Yeah, for us, as I mentioned, that sort of February late cold snap that came through and created that sort of large swath of thin young ice, that was unique for us from what we'd seen in that region and during these cruises, these April cruises in the past. So that was probably the biggest change and difference from that. When we were preparing for this cruise, we were seeing what you were seeing. We were seeing the ice disappearing rapidly from the area along the coast all the way up to Nome. And we were very worried, as I assume you guys might have been, or very wondering what our cruise was going to be like, because it started to look a lot similar, quite similar to the way 2018 was looking, which at that time was our lowest ice year. And then it was a little bit after that we had that cold snap south. There was still that opening area, that sort of Polinia opening just to the west of the coast. But we never got to go up into that area because we didn't have access. So for us, the biggest change was the ice technically ended up being a little bit further south than we normally are in April. The ice was much thinner, very different habitat. And that either for that reason or just because of the increased number of storms, the swells that we were experiencing were much bigger. We also had a lot more fog and a lot more snow. So if it was anything, it was the environmental change was the biggest.
Yeah, thank you. Because the sea surface temperatures to the south of the ice have kind of remained high throughout the winter for the southern Bering Sea. So it's interesting, not surprising you see fog, but the thin ice, thank you, that's also interesting. There was a question in the chat box, but I think maybe Josh London's answering it. It was, did measuring the statistician, the fellow on the ground, did that prove the method is viable? And Josh, I think you just answered that. Maybe you can blurt it out to us.
Yeah, well, and then Mike can tell me if I'm wrong or right. But no, we just started kind of analyzing some of that data, so we haven't really had a chance to look at some of the specifics. But the one thing that measuring the statistician allowed is for Mike and Heather and Gavin, who were the UAS pilots, to really kind of get some extra practice with the drone and to get the sensors dialed in, so that when we had a chance to fly over seals, we had everything working as best as we could.
I would say there were two reasons for doing that. One was we used a laser rangefinder that's attached to the UAS to give us exact altitude or distance to the subject. And we were a little bit unsure as to how well that laser rangefinder was going to work over snow. So we wanted to be able to test that on as many different things as we could. And Paul, our statistician, was one more thing that we could measure. The second thing is, it's always great to bring your statistician out into the field with you. If for no other reason, it gives them an appreciation for just how difficult it is to capture an animal or to collect any data. And so oftentimes statisticians who don't go into the field just get the data back and work it up, and it's quick. It's great to be able to have statisticians out to recognize just how complicated and difficult an endeavor it is to collect this work. And we were lucky that Paul was such a great person to have in the field too. So we're doubly lucky with him.
All right. So there is another question, and there's some about who is your photographer and the quality of the photos and whatnot. But I think the real question is, how long in time wise are those 600-meter ribbon seal dives?
Yeah, that's a great question. The answer is that most of them are in that 12 to 15 minute range. Maybe some of them go up to about 20 minutes. The thing that I've noticed, at least in my kind of general look at both ribbon and spotted seals, is that their dive duration tends to be in that 10 to 12 minute kind of range for most of their dives. And so sometimes when they're diving shorter, they just spend more time down at the bottom. And then when they're diving deeper, they take a little bit more time to get down to those depths and back up. But it's still pretty amazing to me that an animal can make it down 600 meters, 500 meters, 400 meters, those depths, do what it needs to do, and then get back up in 20 minutes or so. It's pretty impressive. But yeah, that's at least from my understanding, that's kind of typical of most kind of smaller seals. We don't see these, elephant seals have much longer dives, bigger animals, longer dives. But for our smaller seals, we tend to see dive durations in that 10 to 15 kind of minute range.
And Josh, my question due to that from that is, does the bladder that ribbon seals have on their chest, on their sternum, that really no one seems to know the actual usage of that, is that something that is taken into consideration as far as somehow allowing a small bodied animal like that to somehow push it at depth?
Not that I'm aware of. I think, one of the things that we get insights when it comes to how ribbon seals are able to do that is their blood, the information that Heather was talking about. The color of ribbon seal blood is deep dark. For those who know blood values, we regularly see hematocrit values in the 60s, upper 60s for ribbon seals. Whereas spotted seals, it's more likely in the 40s or so. And like Colleen says, it's like syrup. There's just a lot of red blood cells in those, and they're dense, and that allows them to store more oxygen. So that's one of the things for ribbon seals that allows them to do those deeper, longer dives. But I've not heard anything about that bladder being related to diving.
Right. All right. Peter Bovang says ribbon seals have high hemoglobin in blood and myoglobin in the muscles. Both are part of the oxygen storage capacity.
Yep.
Yeah, if you've got good oxygen grabbing blood, then put a lot on board. It's good. And for Heather, I've got a question on the pathogens. So out here, you know, COVID for the UME, actually, when the UME on the seals, all the young ones were stranding back in 2018 and 19, that was the time, about the time 20, I think we were getting on board with the declaring it a UME and getting organized. The tiger in the Bronx Zoo got the COVID. And we have a large investigative team, and they said, well, if the tiger keeper can give a tiger COVID, could the seals get COVID? And the seals are being tested now for COVID. And that was out of the UME, and I think that's now a national program. And the other thing that we are now seeing is that avian influenza has arrived into wild birds and domesticated fowl in Alaska. And the only marine mammal that I'm aware of that has ever gotten a little bit of the avian influenza is the harbor seal. And so we're just wondering, are you testing for those two pathogens as well as all the other diseases?
We have tested for influenza A. I don't know what specific, if that's a specific avian influenza. Josh or Colleen might know better. And we haven't found any, we found there's been some positive serology, so positive antibodies, but no DNA, because we've used tested nasal swabs for influenza, and those have all been negative.
Okay, that's great. And that is something that's nice for the region to know up here, because, you know, it's another thing we having to put on our plate as far as concerns. So
Yeah.
Thank you for that. And let's see. Peter Lowry has a.
That was literally my question, Gay. You took it from me.
Oh, I'm sorry. Great minds think alike. Oh, okay. So there it is. The red fox and do marine mammals ever get avian flu? Yes, they can get. They are often tested for influenza A. So thank you, Colleen. And my other question, or any other questions? Okay, I could go all night, but we are going all night, so I better round it off. My question is, oh, we're getting in the chat here. We collected samples to test for SARS-CoV-2 as part of a national effort. Thank you, to screen a range of wildlife populations in coordination with the state of Alaska and the USGS National Wildlife Health Center. Great, thank you. My last question is, when do we get you back? Seems like you guys have, just did 3 straight sciences, I think, tonight. And it looks like I'd love to hear the results of the tagging, and Heather to talk about the pathogens, and Michael to talk about the drones. So we hope to see you all back sometime in the near new year, if not this winter, to sort of give us even more of what you found on your cruise. We really appreciate you coming, and you did a great job. And if you guys want to stay while everyone else signs out, that would be great, just for a second. And last but not least, the reason that Straight Science was on a Wednesday tonight is because commencement at Northwest Campus at Old St. Joe's starts at 7 o'clock tomorrow. And there will be no public, but this year the graduates are allowed to cook I won't cut you off, Peter. I'll bring it around. He has a question for you. The graduates have invitees, so you'll see people at the church, but the public isn't allowed in. And next week's Straight Science will also be on a Wednesday, a little unusual, and that will be about sea ice radar. So NOAA is about to get a sea ice radar set up. And just like they have in Utqiagvik, what that means, the public access that we'll have to it, and what it can tell us, that will be next week on the 18th, Wednesday night. So with that, Peter had a question, so I'm going to swing it back now. One more question for Josh about battery life. Josh.
Yeah,
Peter?
I think,
Peter.
yeah, Peter was asking a
your
question about, yeah, he asked the question in the chat about battery life. And I realized I might have said some contradictory things. And I said that the batteries are limited, but they also last for years. So the batteries are limited in the sense that we have a certain number of transmissions and a certain number of things that the tag can do before the battery dies. But we can program the tag to be less greedy, as Mike would say, about things, and so we can make it last longer. So the tags that we attach to that we glue on, where maybe I only expect the tag to last a month or two, those tags, we let them transmit as much as possible because we want to get as much information as we can right away before the tag falls off. But for the smaller flipper tags that we attach, those only transmit when the seal's hauled out, and they just transmit a few transmissions and then they shut up. Just enough to kind of check in, give us some data, and let us know that where the seal is and what it's doing. But then it may be, weeks or in some cases months before we hear from that seal again. A little bit more trickles out, and by programming it that way, we can extend the life of the battery so that we get years out of it. So the battery life is limited, but we are able to extend the life by programming it in a way that reduces the amount of transmissions.
Okay, Peter got it. Rock on. With that, thank you all for coming. We'll see you next week.